Uh, Proteins are essential part of the cellular machinery, and a protein interacts with other proteins using multiple interfaces. It can also use the same interface to interact with other proteins, with some of them with low binding affinity and with some of them with high binding affinity. A point mutation is a substitution of one amino acid by another one, and disease-related mutations are preferentially localized on protein interaction surfaces. They affect the strength and the propensity with which two protein interacts, and we quantify these effects of the mutations as a changes of the binding affinity, delta delta G of bind. So here I will show one example of the mutation. We have a protein complex, and here I highlighted tryptophan at the interface. And now I mutate tryptophan. It's a missense mutation, point mutation, to glutamic acid. And by doing this mutation, not only the geometrical properties of the interface changed, also the distribution of charges on the interface is influenced by the mutation. So to measure the effects of the mutations on protein-protein interactions using the computational approach, one way is to calculate the binding affinity before and after mutations. We followed this technique by identifying fine descriptors on the interaction site, on interaction surface of the protein. Initially we had some good results, some interesting results. These fine descriptors, they play important role in the prediction of the binding affinity. So here we have a protein complex, and which goes through 30 different mutations, point mutations at the x-axis. And the blue curve represents the binding affinity of each of these mutations, which is measured experimentally. And the red curve shows the predictions of the binding affinity. And we observed that our predictions follows the trend on the blue curves, like following the ups and downs of the binding affinity on the blue curves. So at the beginning we were happy, but we observed that we didn't get a good result for other protein complexes. And in fact, a prediction of the binding affinity is a very challenging task by its own. And if we do some errors in the calculation of the binding affinity, in the estimation of the binding affinity, these errors will be propagated to the estimation of the changes of the binding affinity. Also, we were doing some unnecessary calculations related to the properties that are not modified by the mutations. For example, the chemical composition of the non-interacting surfaces are not changed by the mutation that happens on the interface. So how about calculating the changes of the binding affinity directly using machine learning algorithms? For this, we can rely on both structure and sequence-based information. There are some approaches that use one of them or both of them. Then we developed a deep learning-based framework to capture the local changes caused by the mutations that happens on the interaction surface, on the interface, and try to estimate directly the changes of the binding affinity. In this path, we leveraged the knowledge that exists in the protein-protein interactions in the PDB and transferred this knowledge toward the problems or the tasks related to the protein interactions. To do so, we adapted an approach called ornate, in which a protein, a residue, and its environment are represented using locally oriented cubes. These cubes are centered and oriented based on the common scaffold of the backbone of the central residue. And thanks to this local orientation, we can have a rotation and translation invariant representation. Also, we took into account the different structural regions of the interface, which are support, core, and ream. Here, they are shown with the different colors. Support are the interfacial residues that they are buried on the interface for the monomers, and they become more buried upon the formation of the complex. Core are the interfacial residues that they have solvent accessibility on the monomers, and they become buried upon the formation of the complex. And rim, there are residues that they have solvent accessibility before and after before and after the formation of the complex. So our approach called deep local analysis, and the first part of our algorithm is called SSDLA, which stands for self-supervised deep local analysis, is a structure-based general purpose model to generate informative representation that can help us in the downstream tasks concerning protein-protein interactions. So the goal of this model is to identify or find the residue-specific patterns from the environment around the residue. And we use the masking techniques and self-supervised techniques to build this model. Here is an example of the two examples of the same interface. We have arginine and we have isoleucine, and we select the atoms inside the spheric volume of radius 5 angstrom, randomly centered on the central residue. We select this and we mask all these atoms. We chose this type of selection and masking to avoid any residue-specific biases in our training. So, to train our model, we created a database of non-redundant interfaces we extracted from PDB. And we processed each of these interfaces, all the interfacial residues of each of these interfaces. Here is an example of one of the interfacial residues. The central residue is shown by pink and its environment is in yellow. We mask it here and it goes through a series of 3D convolutional neural networks. And the task for the SSDLA is to predict the masked amino acid. And in this path, after training, we can extract some embeddings from the model that can help us in the PPI-related tasks. So then we wanted to evaluate the performance of SSDLA to see if we can learn a residue from its environment. We created a database of 34,000 interfacial residues. And here is our performance. X-axis are the target, the true amino acids, and the y-axis are the propensity of the predicted amino acids divided into different structural regions, support, core, and ream. Also, we colored the amino acid based on their physicochemical properties. We classified them into 7 classes that you can see them here. So we observed that SSDLA can perform very well for example, for phenylalanine, histidine, tryptophan, and tyrosine, and for also it performed well in general for the aromatic class. For the methionine also it performs well, but for the aliphatic hydrophobic class, it has a little bit challenge to find the residue-specific patterns from the environment. For example, for valine, in the rim region, it cannot find any residue-specific pattern, but in the support and core, at least it finds some patterns coming from the same group, same aliphatic hydrophobic group. Here, the red ones. About the group of hydroxyl containing an alanine, which are shown by black here, besides cysteine, it cannot find the residue-specific patterns in none of the structural regions. Then we performed a simple transfer learning using the frozen embeddings extractor from SSDLA, and we trained a very shallow neural network with only one hidden layer. And the task was to do multi-class classification to find the class of the amino acid, the masked from the mask environment. And we observed the similarity between these tasks and our previous evaluation, particularly for aromatic class, positively charged amino acid, polar and negatively charged, and hydroxyl containing and alanine classes. So now we want to use SSDLA for our major downstream task, which is the prediction of the effects of mutations. And we start with the local environment of the wild type and mutant amino acids. And we use the SSDLA model in a Siamese architecture in the shared weight architecture that you can see here. One branch for the mutant and the other branch for the wild type. And the output of these architecture is the changes of the binding affinity. In this path, we use the mutational landscape of a monomeric protein also as auxiliary features. In addition to that, we use the structural regions, evolutionary conservation, physicochemical properties, and circular variants of the wild-type amino acid. The database that we used for the train and evaluation is SCMP. It has over 7,000 samples. We chose 2003 single point mutations associated to 142 complexes. And for them, we estimated the structure of the mutant complex using Rosetta backdrop modeling. Then for our initial evaluation, we performed tenfold cross-validation, and we reached to the Pearson correlation of 81%. However, this type of evaluation has a little problem. We might have the same mutational position in different folds. To make the better test, we split our data set into train and test, and there is no common complex between these two sets. Our test set has 391 mutations associated to 32 complexes. Here you can see the result. We reached to the Pearson correlation of 73%. This is the exactly same plot, but the samples, the mutations, are colored based on different properties, such as structural regions of the mutation, biological function of the complex, change in the size between the wild type and mutant residue, and sequence identity between the complexes in the test set and the train set. For the structural region, we can see that the overall model performs well for the core and rim regions, although the majority of the samples here are composed of the mutations that are happening in these regions. And before, in our previous research, we observed the importance of the residues belonging to core and rim in the formation of the protein complex. For the biological function, we can see that the machine performs well for the functional class of protease inhibitor, but here we have a little bit data entanglement. In fact, the majority of the samples mutations here, they belong to the protease inhibitor, the orange one, and majority of those mutations are to non-alanine substitutions. The machine didn't perform well for the antibody antigens or TCR major histocompatibility complexes. But this might be due to the fact that the majority of the mutations for these complexes are two alanine mutations. The model showed robustness in the change in the size and the sequence identity between the complexes in the train and test set. We compared the performance with other approaches, including FoldX, Bind, Profix, IC, and MCSM. And overall, the performance was improved on the DLA mutation. As a final test, we compared the performance with IC. IC is a well-known approach to predict the changes of the binding affinity. And it used both the structural and sequence information. They trained on the first version of SCMP and tested on the second version of SCMP. They reached the Pearson correlation of 25%. We followed exactly the same experimental protocol, experimental setting as them, and we reached to the Pearson correlation of 42%. So you can access DLA mutation, the data, and all the models using this link here. So DLA mutation can handle single point mutations, but it doesn't cover the multiple point mutations. Also, we are wondering, we want to study the contribution of conformational sampling, this Rosetta backdrop modeling that we performed in the performance and predictions of the DLA mutation, or alleviate the need to create these mutant protein complexes, or at least make it faster. And if we can answer to this question, we can go toward the systematic assessment of mutational outcomes on protein-protein interactions at a protein-wide scale. At the end, I would like to thank and acknowledge Sorbonne Université, ECCB, and the supercomputer of Jean Zay for all the computation facilities and computational resources. And also, I would like to thank my supervisors, Alessandra and Elodie, for all their help and supervision. If you are interested to join the team, there is a ERC-funded project in the team. And thanks to this ERC-funded project, we have 2 postdoc positions. And the deadline is end of November, so you can scan these codes. Thank you.
We have time for questions.
Thank you. Really nice talk. I used to work with tandem protein repeats, and I was wondering, because I think all the ones you were showing at the beginning were protein tandem repeats, maybe uncurrent. And I was wondering,
which slide?
Right at the very beginning, you had some structures. Yeah, that next one, please. No, when you've got multiple gray ones, you know, with the solenoid domains.
This one.
Yeah, I think those are protein tandem repeats. I was wondering if you had looked at them specifically, because they have all of the, you know, they have these huge interfaces, all of the different copies one after the other interacting with the substrates, and I thought they might be a bit different than just globular domains binding other globular domains.
No, this is only an example. This is only an example. I highlighted the part of the interfaces and pink and the point mutations are blue to give you some visual perceptions of the methodology for the prediction of binding affinity before and after mutations.
Okay, and how do you do the homology, the sequence identity filtering? Because you said you remove redundancy.
So at our complex-based split here, we didn't care about the similarity sequence identity, but during the testing here, we filtered the samples in the test set based on their similarity for both partners between the test set and the train set. And we saw the robustness. And this can be explained by the fact that we focus on the local changes. And these local changes might not be affected by the similarity between the train and test set.
Okay, thank you.
We have time for one more question.
Thank you very much first for your very interesting presentation. So I would have one question. You said that you have taken these delta G values from some kind of database. And I would be very interested in what is the experimental basis for these delta G values? And
Yes.
is it one basis or is it from different experimental setups?
Very good question. There are different experimental methods. Actually, the values were provided by the dissociation constants for the wild type and the mutant, and the experimental methods were various. We chose the four most reliable experimental methods: SPR, ITC, fluorescence, and spectroscopy.
Okay, and because would you expect that I mean, if I measure this once with ITC and then another time with SPR, I would not necessarily expect to get the same dissociation constant.
Correct.
So what would you expect is the correlation there? And would that limit the correlation that you could achieve with your model then?
So this is one of the inherent problems of we face with the database, and we need to study this part, but there are also other issues. For example, if it's the same SPR, we observe different measure for the binding affinity, which need to be addressed.
So thank you, great work. And I think, as you know, the SCAMP2 dataset is very unbalanced in terms of mutation type, mutation position, and so on.
Yes.
And so what could happen is then when you perform tenfold cross-validation, you could obtain like over-optimistic results on this cross-validation because you can learn this type of imbalances. And so have you an estimation whether your method could be extended and still perform as well on more systematic data sets, for example, when the mutation type are balanced, where the location is balanced? And for example, there is a few data sets of deep mutagenesis on one protein, then they tested all possible mutation and stuff like this.
Yes, so yes, exactly. We have these unbalances, and both for example, structural regions where mutation happens, or toward what type of mutation. Like we have a lot to alanine. We haven't studied this, but so basically this tenfold cross-validation, I would avoid to do it because it's very difficult to handle, to make it very fair split between the folds. One thing we can do, we can repeat this complex-based split that we did, repeat it for many times, and to remove any effects of the imbalance in the between the train and test set.